For over 50 years, the Nobel Conference at Gustavus Adolphus College has hosted preeminent scientists, theologians, and ethicists to discuss questions at the intersection of science and society. From the newest results in physics, chemistry, and biology to the newest fields of multidisciplinary study, scientists at the Nobel Conference have examined the universe at its largest and smallest scales, explored the oceans, described new materials, and investigated human and animal behavior. Conference speakers have debated the mechanisms of aging as well as the science and economics of food. Often, speakers have given us a glimpse of the next defining questions and how they might be answered. Throughout all of the conversations, ethicists and theologians have grounded the science in a human dimension. This year's conference focuses on addiction, a uniquely human condition. The 51st Nobel Conference, Addiction, Exploring the Science and Experience of an Equal Opportunity Condition, brings together experts in medicine, neuroscience, sociology, and philosophy to explore the science and experience of addiction. The conference explores a range of questions. What does it mean to be addicted? Is it a brain condition, a psychological disorder, a sociological problem? The answers to these questions raise a very important one. What treatment options are available? We hope you enjoy this year's presentation by Dr. Eric Kandel titled, We Are What We Remember, Memory and Age-Related Memory Disorders. Eric R. Kandel is a university professor at Columbia. In addition, he is the Kavli Professor and Director of the Kavli Institute for Brain Science, the co-director of the Mortimer B. Zuckerman Mind-Brain Behavior Institute, and a senior investigator at the Howard Hughes Medical Institute. Dr. Kandel is internationally recognized for his research on memory. Recently, he has studied animal models in mice of memory disorders, mental illness, and nicotine addiction. Dr. Kandel is the recipient of the 2000 Nobel Prize in Physiology or Medicine.
Thank you, Mike Ferragamo, for that superb introduction. I'm very glad that my wife, Denise, is here to listen to it. I'm really sorry that my parents aren't. My father would have been proud, and my mother would have believed it. I'm delighted to be here, and I'm particularly grateful to Sharon Singh, to Clay Michelson, and to Anna Hollander for their wonderful hosting of Denise and my visit. Today, I'm going to talk to you at this 51st Nobel Symposium and outline for you our current thinking on learning and memory as a background to Denise Kandel's lecture on addiction as a learning process. In my lecture this morning, I'd like to make 3 points. First, that there are 2 major forms of learning that are quite different in their function in your life, implicit and explicit, and I will explain those to you later. Second, that to a large degree, memory makes you who you are. When you remember something, it changes your brain. And third, that memory often declines with age, and we're beginning to learn what you might do to prevent that. Let me introduce learning and memory, 2 of the most magical capabilities of our mind. Learning is the process whereby we acquire new information of the world, and memory is the process whereby we retain that knowledge over time. Most of the knowledge we have about the world, we have learned. It is not built into our brain. As a result of this knowledge over a lifetime, we are in good measure who we are because of what we learn and what we remember. Moreover, specific disorders of learning and disturbances of memory haunt the developing infant as well as the mature adult. Mental retardation affects the quality of life for young people, while normal weakening of memory with age and devastation of Alzheimer's disease haunt the elderly. Memory is the glue that holds your mental life together. Without the unifying forces of memory, our consciousness would be broken up into as many fragments as there are seconds in any day. A life without memory is empty and meaningless. Imagine a life without memory. Clive Waring, a conductor, suffered from herpes simplex encephalitis, which damaged his hippocampus and caused him to lose a particular component of memory, explicit memory storage. Here's Clive Waring. Is the audio on? I don't think the audio is on. Can we put the audio on, please? Thank you.
How many years have I been ill?
About 20.
About 20. Can you imagine what to have one night 20 years long with no dream? That's what it's been like. Just like death. No difference between day and night, no thoughts at all. In that sense, it's been totally painless. Which is not something which is very desirable, really, is it? Because it's precisely like death. If you have no senses of pain, you have no sense of any kind working either. I don't remember sitting down on this chair, for example, or the settee as it is. That was unknown to me. I've never seen a human being since I've been there. That's the first photograph I've seen of anybody.
And who is that photograph?
It's one of my sons. I can't remember his name.
Tragic. The study of memory can be divided into two parts: where in the brain is memory stored, and two, how is memory stored? What are the molecular mechanisms underlying memory storage? Let me begin with the first part: where in the brain is memory stored? This is part of a larger question: can any mental process be localized to a specific region of the brain? And the attempt to answer this question led to some of the greatest intellectual accomplishments in brain science in the 19th century. It all began with an anatomist who worked in Vienna for much of his creative life, Franz Joseph Gal. He provided the first two insights into the relationship between mind and brain. He was the first major person to argue that all mental functions are biological. They arise from the brain. There is no dualism. Moreover, different mental functions, seeing, hearing, smelling, tasting, experiencing things, are localized to different regions of the cerebral cortex. Now, he knew that the cerebral cortex was divided into four major lobes: frontal, temporal, parietal, and occipital, but he realized that these four subdivisions were inadequate to explain the 35, 40-odd mental functions that psychologists had spoken about as being quite discrete. So he used another mechanism for localizing them. He divided the brain into 36-odd regions according to certain functions. The cognitive functions were in the front of the brain. These were causality, comparison, sense of time, sense of tune. The sentiments, sublimity, hope, acquisitiveness, ideality, were in the middle of the brain. And the instinctive function, amativeness, romantic love, collaboration, conjugal love, these are the back of the brain. Now, how did he decide to localize these functions? What tool did he use? Well, he thought that if you use a particular function a lot, that part of the brain grows. And as that part of the brain grows, it pushes on the skull that it underlies. So it pushes on the overlying skull and makes that part more prominent. So, for example, he was struck by the fact that at his class in Gustavus Adolphus, the people who were very bright had a very profound frontal bone, while those that were less gifted had sort of a more shallow frontal cortex. And that's because mental functions such as comparison, ability to tell causality, were at the front of the brain, and as they used them in class, that part of the brain grew and it pushed on the frontal lobe in front of it. He thought that romantic love was localized to the back of the brain because the women in the class that were very romantic, like the two ladies that are hosting Denise and me, they have very prominent backs of the head, while the less romantic ladies had less prominent. And that's because the area of emotiveness, romantic love, is at the back of the brain. And as they manifest that charm, that grows and it pushes on the back of the brain. I cannot describe to you what an impact this made on people, intellectuals throughout the world, particularly in America. You can imagine this caught on. People would not date one another without first palpating the other person's brain. Marriage was impossible without a detailed examination of the skull. It was just marvelous, and everyone thought this was essentially the solution to the localization of function. But there are always skeptics in the crowd, and the French neurologist Marie-Jean-Pierre Florin said, this is nonsense. I don't believe in localization of function. And what experiments did he run? He just did a bunch of correlations. We know how wrong correlations can be. He said, what I'm going to do is I'm going to take this area of emotiveness in experimental animals, and I'm going to eliminate them and see how it affects romantic love. So he got rid of the emotiveness area, and he did this in cats and dogs. They had difficulty walking, but they had no difficulty in making love whatsoever. So he ended up by saying all perceptions, all volitions occupy the same seat in these cerebral organs. The faculty of perceiving, of conceiving, of willing, what merely constitutes, therefore, a faculty which is essentially one. Mental functions cannot be localized to specific regions of the brain. Very strong statement, which again carried the day for a while. But then a giant came along, Pierre Broca. He was a neurologist, and he argued that it's likely that functions can be localized, but we have to use a different approach. I had thought that if there were a phrenological science, it would be a phrenology of convolutions of the brain itself, not on the bumps of the head. So he thought mental functions can be localized to specific regions, but we need another approach to do it. And he came across a patient that provided him a handle on this problem. He encountered a patient who could understand language perfectly well, but could not express himself in language. When that patient died, came to autopsy, he had a lesion in the front of the brain. So Broca wanted to speak to people about it. He wanted to speak at major conferences, and he wanted to have a way of referring to it. And he looked and looked for a name, and he finally decided, in all modesty, the only thing he could do is name it after himself. So he called it Broca's area. He then found 7 other patients that had the same difficulty. They could understand language perfectly well, but could not express themselves in language. When they died, they had a lesion in the frontal lobe, invariably, and invariably the lesion was on the left side. And that caused him to enunciate one of the great principles of brain science, nous parlons avec l'hémisphère gauche. We speak with our left hemisphere. This really electrified the scientific community. In Germany, Fitz and Hitzig began to stimulate the surface of the brain electrically in dogs and found that there was a systematic representation of the body surface. The face area is laterally, then the arm area, then the leg area. So there's a systematic representation of your body surface on the brain. And soon thereafter, Karl Wernicke, a German neurologist, just after graduating medical school, made a wonderful additional discovery. He found a patient who was the mirror opposite of Broca's patient. Broca's patient could understand but could not speak. Wernicke's patient could speak but could not understand. When he died and came to autopsy, he had a lesion at the back of the brain. Broca, again, Wernicke, in all modesty, needed to talk about it, and he called it after himself, Wernicke's area. Wernicke now had the profound insight. He said, when you're speaking about a complex function like language, it's not restricted to one area in the brain. It involves several areas that interconnect. It involves a system of areas. And he pointed out how this works for language. So a complex mental function is not localized to a single region, but several. He said, notice that the auditory cortex and the visual cortex, for which one gets input, for example, you speak to somebody and you hear it, or you read something and you record it, both these areas feed into Wernicke's area for the comprehension of language. Moreover, there's a pathway called the arcuate fasciculus that feeds into Broca's area. Moreover, take a look where Broca's area is. You see this motor strip that fits in Hitzig discovered? There's an area of the motor strip that is concerned with the expression, with motor expression of language. And that sits, the vocalization area sits right near Broca's area. In fact, Broca's area connects to it. So he now had a neural system that really could account for language. We hear language, we read language, it feeds into Wernicke's area, passes through the arcuate fasciculus to Broca's area, and Broca's area is close to the vocalization area of the motor strip. Not only did Wernicke provide us with a very coherent neural systematic understanding of language, which to a large degree still is used in emergency rooms when patients come in with a difficulty in language, but he also made a prediction that you can get a third type of aphasia without a lesion to either Wernicke's areas or Broca's area. What sort of a lesion would produce that kind of a defect? Please, one of the students, scream it out. Cutting the arcuate fasciculus, which interconnects Wernicke's areas to Broca's areas, gives you an aphasia, a difficulty to really speak language coherently. You understand language and you speak it, but there's no connection between the two. It's a little bit like a presidential press conference. Information comes in, information comes out, but the two need not necessarily be related. By the middle of the 20th century, many mental processes had been localized to specific regions of the brain. So that raised the question, can memory be localized? And there was a general skepticism that a complex process like memory could be localized to a specific region of the brain. But the person who solved that was Wilder Penfield, a fascinating guy. Wilder Penfield was trained at Columbia. He wanted to be chairman of the department, but somehow he was denied that opportunity. So he went to Montreal, and with support from the Rockefeller Foundation, he set up the Montreal Neurological Institute, in which he used surgery to explore the behavioral function of the human brain. And he realized that the brain itself doesn't have pain receptors, so you can freely stimulate the surface of the brain. He infiltrated a local anesthetic into the scalp, opened the skull, exposed the brain. And he operated primarily on patients who had epilepsy as a result of scar tissue. If you fall and have a serious injury as a result of skating, or if you have a concussion, it can leave you with a scar, and that scar can give rise to epileptic seizures. And the treatment for that is the removal of the scar that gives rise to that, which often is a small scar. But you want to make sure that in removing that scar, you don't attack Broca's area, Wernicke's area, other areas important in the brain. So he stimulated extensively in the surface of the brain before he operated. And he found if you stimulate the visual area, patient would see, visions of all kinds. If you stimulate the auditory, he would hear sounds. But if you stimulate a certain area, memories would come back. Patient would say, I remember the day of my wedding. Or a woman might say, I remember my first child when he was born. This was just amazing. People were really taken back by that. But this was a correlation. It was not absolutely proven until a tragedy occurred. And that is a patient by the name of H.M., we now know his name is Henry Mollison, was knocked over by a bicycle when he was about 12 years old and sustained bilateral temporal lobe lesions. As a result of this, he had severe seizure activity, which was controlled for the longest time. So he was able to finish high school, able to get himself a job, able to work. But after a while, the seizures got so severe that he couldn't continue working. And he presented himself to the surgeon, William Schofield, who was very much influenced by Penfield. And Schofield operated on him and removed the medial temporal lobe on both sides because this lesion was bilateral. Penfield had never done a bilateral removal. As a result of this removal, H.M. was essentially free of seizures for the rest of his life. He died about 3 years ago. But he was left with the most dramatic amnesia, most dramatic memory deficit you could possibly imagine. Schofield was beside himself. He called up Penfield, and Penfield suggested that he send Brenda Milner, a wonderful cognitive psychologist who he'd been working with, to Scoville to at least get the benefit of studying that patient and seeing what went wrong, what does the hippocampus mediate, and to prevent these kinds of catastrophes in the future. And as Brenda Milner studied HM, she was struck with the fact that despite the fact that he had a severe memory loss, a surprising number of capabilities were intact. For example, he had long-term memories for things that occurred prior to surgery. For example, he remembered to speak the English language. His IQ was essentially unchanged. He remembered the trauma, the awkwardness of childhood, etc., etc. So everything prior to the surgery was intact. Moreover, his short-term memory was completely intact. If you gave him a telephone number, 884-5447, he could repeat it. What he was lacking was the ability to take short-term memory and convert it into long-term memory. This is the major function that HM's revealed, that the hippocampus serves to convert short-term memory to long-term memory. Practice makes perfect, but that conversion doesn't work if you don't have a well-functioning hippocampus. She thought for a while, Brenda Milner, that he couldn't learn anything, that this applied to all areas of knowledge. But then, to her astonishment, she made another fantastic discovery. She found that indeed he can learn certain things as well as you and me. For example, if you give him a mirror drawing task, if you ask him to draw the outlines of a star, Leonardo da Vinci-like, by not looking at his hand, not looking at the pencil, but by looking in a mirror, not looking down, looking sideways. When you and I do this, we make a lot of mistakes on the first trial, but in 10 trials on Monday we get progressively better. Tuesday we start off on a better score and we get better still, and Wednesday we're almost perfect. HM does as well as you and me. But if you ask HM, how come you're doing so much better on Wednesday than you did on Monday? He said, what are you talking about? I've never done this before in my life. So clearly he does the task perfectly well, but he doesn't have any memory for it. So that indicated to Brenda Milner for the first time, and Larry Squire developed this extensively, that memory storage is not a unitary faculty of mind, but it exists in two forms. And let me demonstrate this for you.
Clive Waring has one of the worst cases of amnesia in the world.
I know it's like to be dead now. Day and night, the same, blank. No difference between dreams or anything like that. No senses at all. The brain has been totally inactive. No dreams and no thoughts of any kind whatever.
Clive was a renowned conductor living in London when he was struck down by a virus in 1985. Parts of his brain were completely destroyed, including his memory. However, his ability to play music is unaffected.
Do you feel different when you play music?
I've never heard a note since I've been ill. I don't know what it's like to play music when you're unconscious.
You played us some music about 2 minutes ago.
Not known to me. Totally unknown. I've never heard a note yet.
Amazing. So musical skill obviously is requires conscious participation. It's an explicit memory. But once you master it, it's something you can do automatically. True for bicycle riding. When you first learn how to ride a bicycle, you have to tell yourself, put your left foot forward, put your right foot forward. After you've mastered that, if you talk to yourself, you fall off the bicycle. So this made it clear that memory is not a unitary faculty of mind. There are at least two forms. Explicit memory, which is what HM suffered from, also called the clarity. It's the memory for facts and events of people, places, and objects, and involves the medial temporal lobe and the hippocampus, and this requires conscious attention. And this is in contrast to implicit memory, procedural memory, of skills and habits, non-associative and associative forms of learning, classical conditioning, operant conditioning, habituation, sensitization. These are all implicit forms of learning. These involve the amygdala, the cerebellum, reflex pathways, and do not require conscious attention. And you can have a complete knockout of explicit memory storage, but still have very good implicit memory storage. I studied implicit memory storage in the Snail aplasia. And as you can tell at a glance, this is not only a very beautiful animal, but also a highly recognized one. This is the Nobel Prize, and there are many people in the audience, including my wife, who believe that the snail, rather than the investigator, should have gotten the Nobel Prize. But I'm going to focus on explicit memory storage because it's so relevant to what we're talking about here, and show you some aspects of its molecular mechanisms. Explicit memory storage, as we said before, critically involves the hippocampus. And you can see this very nicely. I don't know what it's like here, but in New York, if you ask a cab driver to take you from 110th Street to 160th Street, he'll say, what are you talking about? But in London, you cannot be a taxi driver unless you really know the streets of London. Repeated examinations before you qualify as a taxi driver. When you do imaging on somebody and you ask him a specific route, the hippocampus lights up. And if you drive the taxi for a while, the hippocampus gets larger. So if you want to have a larger hippocampus, go to London and drive a taxi for a while. When you retire from taxi driving, the hippocampus shrinks. So there's a very good lesson in that, which Denise and I are trying to practice. Don't retire. What are the molecular mechanisms underlying explicit memory storage? This is important because these come into play when we're going to consider the learning processes involved in addiction. The hippocampus receives its input from a region called the entorhinal cortex, which feeds directly to a part of the hippocampus called the dentate gyrus, which feeds to the rest of the hippocampus, and also to the hippocampus directly. And you can do in mice various spatial tasks. The hippocampus is particularly specialized for spatial memory, and most memories have a spatial component. And you can do that very easily in mice, and you can show that what happens with learning is that these pathways become strengthened. Nerve cells communicate with each other at points of contact called synapses. As you heard before from Mike, that can undergo changes of strength. I first showed that in aplasia. It also applies in exactly the same way in the hippocampus. When you learn something, certain connections in your brain become strengthened. And that's what happens with spatial memory. So in baseline condition, one neuron connects to another, but there are modulatory inputs in the brain that are recruited by learning processes. For example, dopamine, which also gets recruited for addiction. When you have a single exposure to something, you develop a short-term memory, and that gives you a transient strengthening of synaptic connections that lasts for minutes. That's the mechanism of short-term memory. But if you are exposed to something repeatedly, practice makes perfect. The signal moves into the nucleus, turns on genes, and gives rise to the growth of new synaptic connections. So you have an alteration in gene expression, an actual anatomical change in the brain. So if you remember anything about this lecture tomorrow, and I don't urge this on you, I urge you to forget it, but if perchance you remember it, it is because you will have walked out of this lecture with a different brain than you walked into. And since this lecture is only a small part of what you're going to learn at the 51st Nobel Symposium, imagine how much larger your brain is going to be at the end of these 2 days than it is now. Now, what are the molecular mechanisms of these two phases? Short-term memory is a strengthening of pre-existing synapses due to the activation of pre-existing proteins. So the machinery is there, just gets activated, and it strengthens the synaptic connections. But with repeated stimulation, a signal moves into the nucleus. It's called the cyclic AMP-dependent protein kinase, pKa, and it activates a transcription factor, which you're going to hear about time and time again in my lecture and Denise's, called CREB, cyclic AMP response element binding protein. That recruits other proteins to give rise to the expression of genes and the growth of new synaptic connections. Now, some people get very nervous in this. They say to themselves, there's an alteration in gene expression? What does that mean? I'm going to go home tonight, make love to my partner because we're planning to have a baby. We're going to have a baby, and that poor baby's going to have to remember what I heard, that drivel that I heard in that lecture. You're not to worry about it. This is not a hereditary change. This is a change in gene expression in specific nerve cells of your brain. It doesn't affect the sperm and the egg. You have my permission to do whatever you like tonight. And one of the wonderful things of doing experiments in mice, which my colleagues and I have been working on since 1990, is one can knock out specific genes and see how it affects behavior. So I told you that this is the signal, cyclic AMP-dependent protein kinase going into the nucleus. We can block that signal and see how it affects things. I showed you before, animals learn this task very well. This is what an animal works when its brain is normal. If you block PK, you dramatically interfere with its ability to retain memory. Let me conclude by telling you something about memory in the aging brain. Now, the interesting thing is, when I was a medical student, we practically never spoke about this, and we didn't really have much of a discussion of Alzheimer's disease. Why do you think this is so? People didn't live long enough to have age-related memory loss. In 1900, and we were seeing patients in 1900, went to medical school in the 1950s. Lifespan was 50 years. Now, men, the weaker sex, live 76 years. The stronger sex, women, live 81 years. Alzheimer's disease only makes its appearance in the very late 60s and early 70s. 3% of people in the 70s have Alzheimer's disease, and in the 90s, it's an epidemic. 50% of people have Alzheimer's disease. But as people lived longer, they also lived better. And one began to see that with the weakening of the body, there's also weakening of memory. And it wasn't clear whether this was a separate entity, normal aging, or sometimes called benign senescent forgetfulness, age-related memory loss, or whether this is an early stage of Alzheimer's disease. So my colleagues and I have focused on this problem to see whether we could separate the two. And we did it by comparing three features. One, the onset and progression of the two disorders. Two, the anatomical localization. And three, the molecular defect. Let me begin with the age of onset. Alzheimer's disease, as I showed you, really only begins in the 70s. But one can explore in mice that do not show Alzheimer's disease, is there an age-related memory loss that occurs spontaneously? And we looked and we found indeed it does, and mice live to be 2 years old, and it occurs in midlife in the 40s. And that's when it's thought age-related memory loss begins in people. Begin to have a little difficulty remembering names, et cetera, et cetera, et cetera. And that begins at midlife. Two, Scott Small began to do imaging, and we had known before that Alzheimer's disease begins in the entorhinal cortex, the input stage in the hippocampus. He found that with aging, the defect is in the dentate gyrus. Dentate gyrus is actually part of the hippocampus proper. So that suggested to us that perhaps we can identify the molecular distinction between the two by looking at genes that were specifically altered in the dentate gyrus, but not altered in the entorhinal cortex, and to take people who did not have Alzheimer's disease to see whether or not we could find molecular defects in the dentate gyrus. So we took people at autopsy, people who had died from various causes, and we did ephemetric chips, which allows us to examine 23,000 genes, and we compared the entorhinal cortex of Alzheimer's disease. These people didn't have Alzheimer's disease, but that's the area that's recruited in Alzheimer's disease, to the dentate gyrus. We found 19 transcripts that changed, either went down systematically or went up systematically. And we focused on one because it's so closely connected to CREB, this major transcription factor. And we found that this gene, which is called RBAB48, the messenger RNA goes down, the protein level goes down. In the dentate gyrus, nothing happens in the entorhinal cortex. In fact, nothing happens in any other area of the brain. What is RBAB48? Chromatin, which I will tell you more about in Denise's lecture, is a combination of DNA wrapped around these spools. And these spools are necessary because the length of DNA is so great that it can't fit into the nucleus until you bind it together. And these spools allow you to do that. But they cover up important control regions, and when you recruit Krebs by activating pKa, you also recruit the Krebs binding protein, and you recruit RBAB48, and that acts on the chromatin structure by acetylating it, opens it up, recruits the Tatabox binding protein and the polymerase that allows you to transcribe genes. So you've got a modified chromatin, and RBAB48 is the king for doing that. So what's essentially happening is this key factor, RBAB48, is present in the young dentate gyrus, but it's reduced in age-related memory loss. So humans allow you to do correlations, but in order to get a really good understanding of this, you really have to turn to animal models. So we looked at the mouse to see, does the mouse, when it ages, and I showed you how clearly it ages, that it show a defect in RBAB48. And we went to the dentate gyrus, and sure enough, this is a young mouse, this is an old mouse, very clear deficit in RBAB48. In other areas, such as CA3, CA1, no deficit whatsoever. We can now do a very nice experiment. This is a young mouse, this is an old mouse. Severe deficit in RBAB48. We can take a young mouse, knock out RBAB48, and we can produce that severe deficit. And that caused a major defect in memory storage. So this is a test of memory. This young mouse learns very well, an old mouse has difficulty remembering it. A young mouse learns very well, but if you knock down RBAB48 selectively in the dentate gyrus, severe memory loss. If you take an old mouse, which has a low level of RBAB48 and does badly on these tests, and increase the level of RBAB48, you can rescue memory loss. So this allowed us to provide a summary of the differences we now have between age-related memory loss and Alzheimer's disease. Age-related memory loss begins earlier. Alzheimer's disease begins later. Anatomic localization, dentate gyrus in age-related memory loss, entorhinal cortex in Alzheimer's disease. And finally, the molecular defect is different. Humans show a linear decrease in RBAB48 messenger RNA and protein as a function of age. Old mice have decreased in RBAB48, and young mice with reduced RBAB48 show memory deficits if you knock down RBAB48. Alzheimer's has a difficulty with apolipoproteins, but does not have a difficulty in RBAB48. So let me conclude by a series of experiments that just emerged in the last few months. Are there humoral or hormonal factors that contribute to hippocampal dysfunction that conserve with cognitive aging, and might exercise overcome these blood-borne insufficiencies? In the last few months, a new factor has emerged, that is young bones. It's good to have young bones. Why? Gerald Kaczynski at Columbia, the chairman of genetics, has recently made an interesting discovery. He's found that the bone is an endocrine gland. It releases a hormone called osteocalcin. It acts on the pancreas, it acts on the testes, it acts on muscles, it acts on the ovary and the liver, on the pancreas, I said this before, but it also acts on the brain. So we began to look to see whether or not, in acting in the brain, does it act on the dentate gyrus, and does it affect age-related memory loss? So we looked at osteocalcin injection into the dentate gyrus, and we saw that osteocalcin injection increases the level of pKa and Krebs. So osteocalcin is really critical in controlling pKa, Krebs, and RBAB48. So this suggested that it might be very important for age-related memory loss. So this is a young mouse, and this is an old mouse. This is an old mouse, and if you inject osteocalcin into the dentate gyrus, this is where RBAB48 is decreased, you increase its memory loss. So you take an aged mouse and you can restore its memory deficit by injecting osteocalcin into the dentate gyrus. If you knock out RBAB48, this injection is ineffective. So clearly RBAB48 is the site for which osteocalcin produces its action. Now what happens with aging? With aging, the serum level of osteocalcin goes down. So this might be a contributing factor to why RBAB48 is going down. And if you exercise, the mouse runs, you see that the serum of osteocalcin level goes up. So osteocalcin releases of bone ameliorates age-related memory loss through CREB1 and RBAB48. Because aging is associated with decreasing bone mass, this decrease in osteocalcin could contribute to age-related memory loss. And conversely, this might explain the beneficial effects on cognition in the aged of vigorous exercise, which builds body mass. Since both osteocalcin and the factor carried by young blood through CREB, RBAB48, osteocalcin might be one of the rejuvenating factors carried by young blood. The reversibility of age-related memory loss strengthens the idea that age-related memory loss and Alzheimer's are distinct diseases. Thus, a sound body helps assure a sound mind. Let me just conclude by showing you the colleagues that I've been privileged to participate with and that I was supported by the Howard Hughes Medical Institute. Thank you very much.